Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine for a moment a future where a devastating inherited disease like motor neuron disease could be met head-on with a powerful, kind of coordinated attack.
Hmm.
We're diving into some really exciting new research today that suggests this kind of future might be closer than we think, especially for families affected by a specific genetic mutation in what's called the superoxide dismutase 1 gene.
Yes, today we're taking a deep dive into a fascinating new study from the University of Wollongong. This research looks at combining three existing medications, copper, acetylthiosemide, carbazonate, Ebselen, and Telbivudine,
Right.
as a potential way to treat inherited motor neuron disease that's linked to mutations in the superoxide dismutase 1 protein.
So you flagged this really interesting research, and we're going to unpack it using the details from the scientific paper published in eBiomedicine and also pulling in some info from a recent health news article reporting on these findings.
Exactly. Our goal with this deep dive is to really get to grips with this novel approach of using multiple therapies together, what's being called polytherapy.
Polytherapy. Okay.
We want to understand what the underlying science tells us about its potential and, crucially, what this could mean if you or someone you know is impacted by this particular form of motor neuron disease.
Right.
We're going to try and break down some of the more complex scientific ideas into, clear, understandable pieces, really focusing on those aha moments and making sure we don't get bogged down in unnecessary jargon.
Okay, sounds good. So let's start by laying a bit of groundwork. Talk about motor neuron disease itself.
Sure. So while the majority of cases of motor neuron disease seem to occur, sporadically, without a clear genetic cause.
Right, most cases are like that.
Yeah. But it's important to understand that a significant portion, maybe around 5 to 10%, is actually inherited, passed down through families.
5 to 10%.
And within those inherited cases, a protein called superoxide dismutase 1. It's really important.
Superoxide dismutase 1. Got it.
Normally, this protein acts as a very powerful antioxidant enzyme inside our cells. Think of it like a cellular bodyguard,
Okay, a bodyguard.
Yeah, constantly working to protect our cells from damage caused by these harmful molecules called superoxide radicals.
And in Australia specifically.
Well, in Australia, it's estimated that around 35% of inherited motor neuron disease cases involve changes or mutations in the gene that tells the body how to make this superoxide dismutase 1 protein.
Wow, 35%, that's quite specific to superoxide dismutase 1 here. And I read there are loads of different mutations.
Oh, absolutely. Over 150 different known mutations in that single superoxide dismutase 1 gene.
And what do they actually do at a biological level?
Well, while each mutation is unique, they all tend to lead to a similar kind of problem. They change the structure, and therefore the stability, of the superoxide dismutase 1 protein itself.
Okay, so they mess with the protein's shape.
Exactly. And here's where it gets critical. In motor neurons, those are the nerve cells controlling our muscles. These subtle changes in the protein structure cause the superoxide dismutase 1 proteins to misfold
Misfold.
and then start to clump together.
Aggregate. Clumping. That definitely sounds bad. What happens when these proteins start sticking together inside the motor neurons?
Yeah, it can be quite damaging. These clumps, they interfere with the normal running of the cell in several really critical ways.
How so?
Well, first, they stop the motor neurons from being able to effectively clear out harmful waste products, like a blocked drain, sort of.
Right. Things back up.
And they also disrupt the cell's ability to produce the energy it needs to function properly.
So waste builds up and the power supply gets cut.
Pretty much. And over time, this buildup of toxic proteins and the lack of energy leads to the death of these essential motor neurons.
That's devastating. No, it's so important to remember that science doesn't happen in a vacuum, right? This study builds on previous work.
Absolutely. The study we're really digging into today builds upon years of dedicated work, and that definitely includes the incredibly important contributions of the late Professor Justin Yerbury.
Professor Yerbury.
Yes. Professor Yerbury, who himself actually lived with a superoxide dismutase 1 mutation, dedicated his career to really understanding the precise ways these mutations lead to motor neuron disease.
Wow.
His research was absolutely key in highlighting the fact that the mutated superoxide dismutase 1 protein is inherently unstable and has this very strong tendency to form those toxic clumps we were just discussing.
So he really laid the groundwork for understanding that instability.
He really did. And it's a real testament to his passion and dedication that his research team has continued this vital work after his passing.
Okay, so this new study takes a slightly different approach then. It's not just looking at one single drug.
That's a really crucial point to understand, yes. The major difference in this research is that the scientists investigated using a combination of 3 different drugs that polytherapy approach,
Right, the polytherapy.
rather than following the more traditional route of testing just a single drug on its own.
And what was their main aim with this combination?
Their main goal was pretty ambitious: to see if this specific combination of drugs could help, well, repair the damaged superoxide dismutase 1 protein, stop it from clumping up, and ultimately protect those vulnerable motor neurons.
Okay, repair, prevent clumps, protect cells. Got it. And they picked a specific trio of drugs. Let's talk about them. The study calls the combo CET. The first one is copper acetylthiosemicarbazonate. That's a mouthful.
It certainly is. Maybe we can refer to it as the copper compound for shorthand sometimes. But yes, copper acetylthiosemicarbazonate.
Okay. Copper acetylthiosemicarbazonate. What's its story?
Well, this copper compound has shown some promising results in previous research, especially in lab models using mice that have motor neuron disease linked to the superoxide dismutase 1 protein.
Ah, so it had some history with this.
Yes, and it's also worth noting that it's currently in phase 2 and phase 3 clinical trials for amyotrophic lateral sclerosis, which is, another term for motor neuron disease.
Right, ALS. So it's already being tested in humans.
Correct. And we already have some understanding of how it might work. It seems to help the superoxide dismutase 1 protein fold into its correct shape and reduce the formation of those harmful clumps or aggregates.
Okay, so that's drug number 1. What about the second one, Ebselen?
Ebselen is interesting because it's actually already an approved molecule.
Approved, meaning safe for humans.
Yes, it's been deemed safe for use in humans for treating other medical conditions. Researchers identified that Ebselen seems to reduce the harmful effects of mutated superoxide dismutase 1.
How does it do that?
It specifically encourages the formation of what's called an intramolecular disulfide bond within the protein. You can kind of think of this bond as a tiny internal staple.
Staple.
Yeah, it helps the protein maintain its correct three-dimensional shape. And that correct shape is essential for the protein to function properly, to do its job.
And the study mentioned something about most mutations not affecting the parts needed for this staple, right?
Exactly. That's a key point. The majority of these superoxide dismutase 1 mutations don't actually disrupt the specific building blocks called cysteine residues that are involved in creating that disulfide bond, that staple.
So Ebselen could potentially work for lots of different types of these mutations.
That's the hope. It has the potential to be effective across a wide range of motor neuron diseases caused by different superoxide dismutase 1 mutations because it targets a part of the protein structure often left untouched by the mutation itself.
Okay, that makes sense. And the third drug in the CET mix is Telbivudine. That sounds like an antiviral.
It is, yes.
Yeah.
Telbivudine is an antiviral compound. What's particularly intriguing here is that it was found to lessen damage to axons.
Axons, the nerve cell wires.
That's right, the long projections of nerve cells that transmit signals. It showed this effect in a zebrafish model of motor neuron disease linked to superoxide dismutase 1.
Zebrafish, interesting. Do they know how Telbivudine might work?
Well, the researchers have a hypothesis. They think Telbivudine might work by interacting with a very specific spot on the superoxide dismutase 1 protein, an area called tryptophan 32.
Tryptophan 32.
Yeah, and this spot is thought to play a crucial role in the process of the proteins clumping together, the aggregation process.
So 3 drugs. Copper acetylthiosemicarbazonate helping folding, Ebselen adding a stabilizing staple, and Telbivudine potentially interfering with clumping at a specific site.
That's a good summary of the potential mechanisms, yeah, each tackling a slightly different aspect of the problem.
So how do they actually test this combination? They started in the lab, I assume.
Yes, they started with what are called in vitro experiments, basically. Experiments done in the lab, often using cells grown in dishes.
Cell cultures.
Exactly. They used quite a sophisticated method called a three-dimensional checkerboard assay. Imagine like a grid,
Okay.
where they could test many different concentrations of each of the 3 drugs, both individually and in various combinations, all at the same time.
And they tested this on specific cells.
Yes, on NSC34 cells. These are cells that are very similar to neurons, And crucially, the researchers had modified them to produce large amounts of a specific mutated form of superoxide dismutase 1, known as SOD1-G93A.
Ah, the G93A mutation. That's a common one used in research models, isn't it?
It is, yes. It's a well-established model for studying this type of motor neuron disease.
And what were they looking for in these lab experiments with the checkerboard?
The main goal was to find the sweet spot, the optimal ratio, and the best overall concentration of these three drugs working together.
The combination that best helped the cells survive.
Precisely. The one that best improved the survival of these cells that were churning out that faulty superoxide dismutase 1 protein.
And did it work? Did the combo improve cell survival?
It did, yes. The combination of the three drugs, this CET polytherapy, significantly improved the survival of these cells expressing the mutated protein.
Better than the copper compound alone?
Yes, significantly better when compared to using various concentrations of just the copper acetylthiosuccinate carbonate on its own. That was a really important early finding.
That is important. What about those toxic protein clumps? Did the combination affect them in the lab?
It did. The CET polytherapy proved to be more effective than the lower concentrations of copper acetylthiosuccinate carbonate alone in reducing the formation of those superoxide dismutase 1 protein clumps, also called inclusions inside the cells.
More effective. Okay.
And interestingly, the CET combination was actually as effective as the highest concentration of the copper compound they tested.
As effective as the highest dose.
Yes, but, and this is potentially significant, it achieved this using a lower overall concentration of drugs.
Ah, so potentially fewer side effects down the line if it gets to humans.
Yes, the implication, yes. Getting the same benefit with less total drug exposure is generally a good thing.
Definitely encouraging. Did they look at the protein folding too? Whether the combo helped fix the shape?
They did. The researchers found that the CET treatment increased the proportion of correctly folded superoxide, dismutase 1 protein specifically, the form that has that important disulfide bond, the staple we mentioned.
Okay, more correctly folded protein.
Yes, and this was compared to treatment with just the copper acetylthiosuccinate carbonate alone. It suggests the combination is doing a better job helping the protein achieve its proper functional structure.
And what about the protein's actual job, its enzyme activity? Was that affected?
Good question. Both the CET combination and the copper compound on its own increase the proportion of the superoxide dismutase 1 protein that was in its active form, which is called a dimer, basically two protein molecules joined together.
Active form increased. Good.
And they saw similar levels of superoxide dismutase 1 enzyme activity in both the CET group and the group that got just the single copper drug.
So the treatments boosted the amount of functional protein, not just the total amount.
That seems to be the case, yes. A good sign that the intended effect on protein function was happening.
Okay, so the lab results looked pretty promising, but the real test is often in a living organism, right? Did they move to animal studies?
Yes, they did. They moved on to use a very common animal model for this specific type of motor neuron disease. Mice that have been genetically engineered to carry that same SOD1-G93A mutation.
The G93A mice.
Correct. These mice develop a disease that, mirrors many of the key features we see in the human condition.
And I guess the first thing is always safety and, well, can the drugs actually get where they need to go, like into the brain?
Exactly. That's critical. The researchers first did tests in healthy mice to check the bioavailability of the CET combination.
Bioavailability meaning, do the drugs get absorbed and reach the target?
Yes. Essentially, can the drugs get into the bloodstream and, importantly, can they cross the blood-brain barrier to reach the central nervous system?
Ah, the blood-brain barrier, that tightly controlled gatekeeper for the brain.
Precisely. The brain is this highly protected fortress, and the blood-brain barrier is like a very strict security system controlling what gets in.
So could all three drugs get past it?
Yes. The test showed that all three drugs in the CET combination did indeed reach the brain. That's a significant hurdle clear.
That is huge. And were they safe? Tolerated, okay.
Encouragingly, yes. The combination was generally well tolerated over a 30-day period in these healthy mice. They saw only minimal adverse effects in terms of the mice's weight, their behavior, and standard blood tests.
Okay, good. So safe, gets to the brain. Then they tested it in the G93A mice with the disease to see if it helped.
Yes. They then moved on to the diseased mice. They compared mice getting no treatment, mice treated with just the copper acetylthiosuccinate carbonate, and mice getting the full CET combination therapy.
And what did they find? Did it affect when the disease started?
Yes, that was one of the key findings.
Yeah.
The mice treated with the CET combination showed a significant delay in the onset of disease symptoms.
A delay, great. How do they measure that?
They specifically looked at the appearance of hind leg clasping. It's an early indicator of neurological decline in these mouse models. And this delay in symptom starting was more pronounced in the group getting the CET combination compared to both the untreated mice and those getting only the copper compound.
That's a really positive sign. What about their physical abilities? Motor function?
They tested that too, using something called a rotorod test.
Rotorod? Like a spinning log they have to stay on?
Basically, yes.
Yeah.
Measures coordination and balance. The mice that got the CET treatment performed better on this test.
So improved motor coordination and function.
Compared to the other groups, yes. It suggests the treatment was having a real, tangible benefit on their physical capabilities.
Okay, delayed onset, better function. What about lifespan? That's often a key measure.
It is. And the CET treatment did lead to a modest but statistically significant increase in the lifespan of the mice.
An increase in survival compared to both other groups.
Yes, compared to both the control group and the group treated with just copper acetylthiosemicarbazonate alone.
Okay. Modest but significant.
Interestingly, though, the researchers noted that this survival benefit seemed to be more pronounced in the female mice in the study.
Oh, really? A sex difference? That's fascinating. Why might that be?
That's a great question, and honestly, the study doesn't provide a definitive answer. It hints that there might be underlying biological differences in how the disease progresses, or perhaps how it responds to these specific drugs between males and females. This could be a really crucial area for future research, potentially leading to more tailored treatment approaches down the line.
Absolutely. Worth keeping an eye on. Did they also look inside at the spinal cords at a biological level?
They did, yes. They conducted detailed neuropathological analyses, basically looking at the spinal cord tissue under the microscope and using biochemical tests.
And what did they see there?
They found that the CET treatment led to higher levels of soluble superoxide, dismutase 1 protein in the spinal cord.
Soluble protein. Is that the good kind?
Generally, yes. Soluble protein is usually considered the more functional, less problematic form compared to the aggregated clumps.
Okay, more soluble protein. What else?
They also observed a higher proportion of correctly folded superoxide, dismutase 1 protein in the spinal cords of the CET-treated mice.
More correctly folded protein, too.
Yes, which again supports the idea that the drug combination is helping the protein adopt its proper structure, the one it needs to function.
And crucially, what about the bad stuff? The misfolded toxic protein clumps that are the hallmark of the disease.
This is a really key finding. The CET treatment resulted in a significant reduction in the accumulation of that misfolded superoxide, dismutase 1 protein in the spinal cords.
A reduction in the misfolded stuff, better than the copper compound alone.
Yes, the reduction was more substantial than what they observed in the mice treated with just copper acetylthiosemicarbazonate alone. This directly addresses one of the core problems in the disease.
That sounds really important. And what about the motor neurons themselves? Did the treatment actually protect the nerve cells?
Both the copper compound alone and the CET treatment did show evidence of neuroprotection, meaning they helped preserve motor neurons.
So both helped save neurons?
Yes. The mice treated with the CET combination had a greater number of surviving motor neurons in their spinal cords compared to the untreated control group.
Okay. But was it significantly better than the copper compound alone in saving neurons?
Interestingly, the difference wasn't statistically significant when compared directly to the group that received only the copper acetylthiosemicarbazonate in terms of just the number of surviving neurons.
Okay. So CET was better at reducing misfolded protein, but maybe similar to the single drug in overall neuron count, at least in this study.
That seems to be what this particular analysis showed, yes, though the trend was towards more neurons with CET.
Right. So taking all these findings together, the lab results, the mouse studies, what are the big picture implications here?
Well, I think one of the most significant implications is the strong support this study provides for the whole idea of using a combination of drugs, polytherapy.
Right, attacking the problem from multiple angles.
Exactly. Targeting different aspects of the disease pathology at the same time. In this case, the combo seems to be hitting both the misfolding and the aggregation of superoxide, dismutase 1 protein.
So it suggests this multi-drug approach might be the way forward for complex diseases like this.
I think that's what the research really points towards, yes. For diseases as intricate as motor neuron disease, or maybe even others like Alzheimer's or Parkinson's, the future of treatment might lie not in finding that one single silver bullet,
Yeah.
but in carefully putting together these multi-drug cocktails that attack the disease from multiple weak points. It's kind of a fundamentally different way of thinking about tackling these conditions.
And you mentioned earlier the lab results suggested you might be able to use lower doses in combination.
Yes, the in vitro findings indicated that the CET combination could achieve similar or even better results compared to using higher doses of the copper acetylthiosemicarbazonate on its own.
Which could mean fewer side effects, potentially.
That's the hope, yes. It raises the exciting possibility of potentially reducing the risk of side effects by using lower doses of each individual drug when they're part of a combined therapy.
It really drives home just how complex motor neuron disease is, doesn't it? And why hitting it from different angles might be necessary.
Precisely. Motor neuron disease is so multifaceted. This research really strengthens the idea that a multi-pronged therapeutic approach targeting various underlying biological mechanisms might ultimately be more effective than relying on therapies that focus on just one single target.
And it's encouraging that this isn't starting completely from scratch. These drugs have some history.
Absolutely. This study leverages the existing understanding and potential of these individual molecules. Copper acetylthiosemicarbazonate is in trials. Ebselen is approved for other uses.
Right.
That could potentially help streamline the path towards future clinical trials in humans, maybe make it a bit quicker than developing something totally new.
And for the families listening who are directly affected by inherited motor neuron disease, because of these superoxide dismutase 1 mutations, this must offer a real, glimmer of hope.
I think that's absolutely right. These preclinical findings, while still early stage, provide a genuinely promising new direction for developing treatments specifically for this particular subset of motor neuron disease. It's tangible progress.
Definitely. But of course, with any scientific research, especially early stage like this, we have to talk about the limitations, right? Be realistic.
Oh, absolutely. It's crucial. There are several important limitations to keep in mind here.
Okay, like what?
Well, firstly, and perhaps most importantly, the findings are based on a mouse model of the disease.
Right. Mice aren't humans.
Exactly. While these animal models are incredibly valuable tools for research, they don't perfectly replicate the full complexity of human motor neuron disease. So we always need to be cautious about directly translating these results to people.
Makes sense. And we touched on this earlier, that difference between male and female mice in the survival benefit.
Yes, that's another key point. The survival benefit was more pronounced in the female mice. This suggests there might be sex-specific responses to this particular therapy.
Which needs more investigation.
Definitely. It's an area that really warrants further study to understand why that difference exists. It can have real implications for how treatment is approached in men versus women.
And the study used one specific mutation, didn't it? The SOD1-G93A1.
That's correct. While the SOD1-G93A mutation is common in lab models, it's just one of over 150 known mutations in the superoxide dismutase 1 gene that can cause this form of motor neuron disease.
So we don't know for sure if this CET combo would work for all the other mutations.
Exactly. Further research will be needed to determine if this polytherapy would be equally effective for people with other types of superoxide dismutase 1 mutations. The researchers themselves even noted it might have limited effects on certain types, like truncation variants where big chunks of the protein are missing.
Okay, so its applicability across different mutations needs more research. Any other major limitations?
Well, while the study clearly showed the combination was effective both in the lab and in the mice, the exact way these three drugs work together synergistically.
How they cooperate.
Yeah, the precise molecular mechanisms of that synergy aren't yet fully understood. We see the positive outcome, but the detailed molecular interplay between copper acetylthiosemicarbazonate, Ebselen, and Telbivudine still needs more exploration.
So we know what happened, but not perfectly how it happened.
That's a good way to put it. The researchers themselves suggest future work should specifically look at the individual contributions of Ebselen and Telbivudine within a living organism to better understand how each piece contributes to the overall benefit.
Okay. Now, that health news article we mentioned earlier, it had some comments from the lead investigator, Dr. Jeremy Lum. What was his take?
Yes, Dr. Lum emphasized how novel their approach was testing this drug combination instead of just a single agent. He sounded really quite enthusiastic about the promising results they saw.
Especially regarding disease progression and survival in the mice.
Exactly. Particularly reducing disease progression and increasing survival rates in the mouse model. He seemed genuinely encouraged by those outcomes.
Did he also touch on that idea of tackling the disease from multiple angles?
He did. He highlighted his belief that because motor neuron disease is so complex, a addressing it through multiple biological pathways at the same time, using a combination of drugs, holds greater promise than relying on a single strategy targeting just one thing.
Makes sense. And the article mentioned the connection to Professor Yerbury's legacy.
Yes, that came through strongly. Dr. Lam and the team expressed their deep pride in building upon Professor Yerbury's foundational work and continuing his vital investigations.
Carrying the torch, so to speak.
Yeah, they spoke about how truly inspiring his relentless efforts were and how instrumental his work has been in getting them to this important stage in their own research.
It really highlights the dedication involved in this kind of research. Okay, as we start to wrap up this deep dive, what's the call to action for listeners who've been following along?
Well, firstly, I'd encourage everyone to stay informed. Keep an eye on future research developments in this area. It's a rapidly advancing field. Look out for updates on this specific polytherapy approach and others too.
Good advice. And if people want to get more involved,
Supporting organizations dedicated to funding motor neuron disease research can make a real difference. That funding is critical for driving progress.
absolutely. And if this hits close to home for someone listening, if their family is affected by superoxide dismutase 1-related MND.
Yes, if this resonates personally, we strongly encourage reaching out to relevant support networks and definitely discuss these findings and any questions you have with your own medical professionals.
Right, get personalized advice.
Exactly. They can provide guidance and the most current information relevant to your specific situation.
Okay. And finally, let's leave our listeners with a thought to chew on.
Yeah.
If this three-pronged approach shows such encouraging promise in these early studies, what other combinations could be out there? Combinations of existing drugs or maybe new ones coming through the pipeline? Yeah.
Could they unlock even more effective treatments? Not just for motor neurons disease, but maybe for other complex neurodegenerative conditions too.
It's a big question.
What if the real key to tackling these incredibly tough diseases isn't finding that single magic bullet, but figuring out the right symphony, the right carefully orchestrated combination of therapies, each hitting a different vulnerability?
That's a powerful thought. Thank you for taking this deep dive with us today.
So the main takeaway from our discussion today is that this new research exploring a combination of 3 drugs, copper acetylthiosemicarbazonate, Ebselen, and Telbivudine, has shown significant promise in these preclinical studies for inherited motor neuron disease linked to the superoxide dismutase 1 gene.
Yeah, it really offers a hopeful glimpse towards maybe developing a more effective treatment strategy, hitting multiple parts of the disease process at once.
For those who want to dig deeper, you can look for the published scientific paper in the journal eBiomedicine, and we'd also encourage exploring websites of motor neuron disease research organizations for more info and updates. Absolutely, and this study really does highlight the exciting potential of polytherapy. You know, combining existing drugs in novel ways as maybe a really powerful approach for tackling these complex and challenging diseases in the future.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.